Hello student. In last lecture we have gone through the systems of transmission of that particular type of molecules. We can say the osmosis and diffusion. These two processes we have seen in last few lectures. In today's lecture we are going to start the new topic, that is the plasmolysis. Now it is the term that we called plasmo and plasmolysis. So it depends on the potential of water, and it also depends on the pressure of that water. So on the basis of these two concepts, the osmotic pressure and the diffusion or the osmosis, we can say there will be a different types of situations which occurs in a plant cell or even in a living cell. So what are that situation? Now as we know that there is a different types of solution available for the living organism. Say for example, the solution which is having the pH more than 7, sometimes the solution having the pH less than 4, or we can say less than 7, and sometimes the neutral solution is also there. So there will be a different number of solutions available to the organism in the environment. Say for example, we are looking for the bacterial cell, we are looking for the plant cell. There will be a different conditions where the different solutions are available for the plant. Sometimes the pH of that solution is less than 7, greater than 7, or sometime it will be a 7. So depending on this availability of solution, there will be a three different conditions that we called isotonic solution, hypotonic solution, and hypertonic solution. These are the these are the three different solutions based on the availability of their pH, or it is related to the pH. Now here you can see this one is the parameter, having suppose the beaker, okay, and in the beaker we have added a cell. Say for example, a plant cell, we have added to the water. No water released from cell, no water enter to the cell. So there will be a hardly any exchange of the water molecule. Means not a single drop will come throughout the cell, or not a single drop is added to the cell. So the cell remain as it is in that particular solution. So solution, we can say suppose water. So what we conclude from this? We can say that the potential of the inner side of that cell is having the equal potential of the outer side of the cell. So we are talking about the potential of water. So water potential is having the equal in outer and external side of the cell, then this condition will occur. There is no any flow of water from external side to the inside and from internal side to the external side. So this solution is known as isotonic solution, when the concentration of solution is similar to that of internal cell solution or internal cell concentration. Now if we have added a cell in a solution and cell starts to release the water from inside to outside, so the flow of water will take place from inside to the outside. In this situation we can say the solution is hypertonic. When the water molecule will move from inside to the outer side, then we called it as a hypertonic solution. Now the third condition, if water enter into the cell, so from external side water will move towards the inner side of the cell, then this condition is known as the solution is hypotonic solution. So these are the three conditions where the water movement will change, and depending on the movement change we can say the solution is either isotonic, the solution is either hypotonic, or solution is either hypertonic. So among the three, one solution is there. So there will be a effect of these solutions on the cell. Now here you can see hypertonic solution is there, and in that we have added a cell. When after some times we observe this cell under microscope, it looks like this particular situation as given in A, where the water molecule will move from internal side of the cell towards the outer side of the cell, so that what happens, the cell will become plasmolized. So this type of cell is known as plasmolized cell. Now in second condition, where the isotonic solution is there is no any flow of water from inside to the outside and from outside to the inside, so the cell remain as it is. So it is known as the, suppose a flaccid cell, and it remains as it is for a longer duration. Now the third condition is there, the cell is added to the hypotonic solution, where the cell starts to become a turgid, and it will increase the size, and it will attain the maximum size of that particular cell, and it will starts to burst. So this is nothing but the three condition: plasmolysis, flaccid condition, and turgid condition, depending on the type of solution. Hypertonic solution is there. When we add the cell to the hypertonic solution, that particular cell become plasmolized. When we add the cell to the isotonic solution, the cell remain as it is, because there is no any flow of material or flow of solution or flow of water from inside to the outside. And third condition is there, where the cell starts to become a turgid, and after a particular stage this cell starts to burst, so that we called it as a hypotonic solution. So depending on the type of solution, these three conditions were studied, or we can say these three conditions will occur. Plasmolysis occurs when water moves out of the cell and cell membrane of the plant cell shrink away from its cell wall. So here the shrinkage of the cell or shrinkage of the plasma membrane will take place, and this we called it as a plasmolized cell. On the other hand, when water move inside to the cell become turgid, as it is given in the hypotonic solution. So depending on the solution, these two conditions are most importantly occur. Now the next terminology which is generally used in plant physiology is imbibition or imbibition. Imbibition is a special type of diffusion when water is absorbed by solids that we called colloids, causing them increase in their volume. So absorption water, example we can say absorption of water by seed and dry wood. So when we look at the seed, initially the seeds were dried and they will become a very compact structure. As we add these seeds in a water, they will utilize this water and they will become a turgid, or we can say they will starts to increase their size, and that we called it as a, and this process we called it as a imbibition or imbibition. Now moving to the again our topic, that long distance transport. Again before starting this, we can take a look towards the discussed point. Up till now we have discussed the 4 points. One, we have discussed the diffusion, we have also gone through the osmosis, then we have seen the plasmolysis, and the last terminology which we discussed, that is the imbibition or imbibition. Now these 4 terminologies, or we can say these 4 processes, are the most important phases or processes which takes place in short transport of water molecule. Means within the cell or within 2 cells, the movement of water will take place through these processes, and due to this process we can say that short transportation of water will occur in the plant molecule. Say for example root hairs are there, so the root hairs will consume or absorb the water, then it is given to the cell, and again from this cell it will given to the other cell. Likewise, cell to cell movement or within a two cell movement of water molecule will takes place, and this will done with the help of osmosis, with the help of diffusion, with the help of imbibition, with the help of plasmolysis. So these are the processes depending on the different types of situation. Say for example, concentration of water is also important thing in this particular processes. Then the water potential is important, then solute structure is important, then solution type is important. So all these different types is having the influence on these processes that we call osmosis, diffusion, plasmolysis and imbibition. So here we stop with the discussion of these four types.